aerosols that require additional protection, that do not have any FDA approved intervention strategies. There is a risk of transmission. So those sorts of pathogens where we have to be very careful in how we deal with the pathogens, we work with them in the BSL-3 environment. So my lab does both cell culture based and animal based work with BSL-2 and BSL-3 pathogens. Next slide, Laura, please. So as I said, we have been doing this since, I don't even remember when we started. I've been doing virology work for as long as I remember, I think. We work with many acute viruses. What this means is that these viruses come in, they cause an infection, they cause disease, and they leave. So things like influenza, right now, what is happening in some ways, although it's still in the fuzzy area, is SARS-CoV-2. These are all short-term disease situations. So we work with a lot of those sorts of viruses, many different viruses. Many of these things you might not even have heard of them. They remain, thankfully so, exotic to some people, just because they are not so prevalent. We work with many viruses that cause encephalitis. We work with viruses that cause hemorrhagic fevers during late stages of infection. Many of these viruses are transmitted by mosquitoes, but many of them are also transmitted as aerosols. And as aerosols, they cause significant disease. So what is not included in this list, but what we have been spending most of our time with since early last year, is working on SARS-CoV-2. Again, because we have a BSL-3 lab, and working with SARS-CoV-2 requires BSL-3. In all of these cases, we say, how do the virus and the pathogen, how do the virus and the host communicate with one another? What is that language that they use to communicate with one another? Can you use that information to better understand what the virus actually needs to cause disease? And can you take those away? In other words, can you identify drug targets? Can you use FDA approved inhibitors, things that have already been approved for other indications? Can you use them to treat these conditions? That's called drug repurposing. We do a lot of that. We pay a lot of attention to inflammation, because in all of these cases, in every single one of these viruses, inflammation is an integral component that contributes to disease. It is the evil twin. So we look at inflammation in all of these things, and we have programs that look at vaccines in all of these cases. Next slide, please. And we do these in different model systems. So there is a pipeline, so to speak. You do a lot of work in cell culture first, and you identify things that make sense. Then you have to transition them into larger, more relevant models so that you can actually understand how a pathogen is controlled by a given therapeutic, how efficacious is your vaccine strategy. So we have small animal models for many of these things. These animal models also include exposure, treatment, vaccinations. All of these things are, kind of roped into these models. In addition to that, one cool thing that we have here is organ on chip platforms. So these are human-derived organs or representations of organs on chips. These are small glass chips or glass slides, and you have multicellular tissue environments reproduced in those glass slides. And so you can actually do some of these validation studies for these various candidates in a human microenvironment. And then moving on to the next slide. So we have all of these things that are in the pipeline. So this is going, all of these things are in various stages: cell culture, animal models, organs on chips. Right now, we have organs on chips for the brain and for the lung. We are looking to expand that to the heart, probably circulation and blood vessels very, very soon. Potentially the liver coming down the pike, but right now the big ones are the brain and the lung. We have multiple therapeutics that are going through the pipeline for many of these things. Many of them are FDA approved already. So we have a significant effort in drug repurposing. We also do a lot of work with peptides. These are small peptides, small amino acid, multi-amino acid chains, which have antiviral activity. But the cool thing about them is many of them are inherently immunomodulatory. So you can kind of get 2-for-1, buy one, get one free sort of a situation where you can have the same agent control the virus and also inflammation. We're also looking at biologics, many different kinds of antibodies, plenty of work ongoing with vaccines, many of them focusing on delivery platforms, DNA-based delivery platforms, protein-based delivery platforms, lipid-based platforms. And from a basic science perspective, we are also looking at what your immune system is seeing when a cell is infected by a pathogen. That is absolutely critical for you to be able to design a good vaccine and make sure that the vaccine is also presenting the same thing to your immune system. So those are all the kinds of things that are ongoing right now. So I think that is my last slide. With that, I will stop.
Wow, wonderful. Thank you so much. I forgot to mention in the beginning, if anyone has any questions for any of our faculty, please put it in the chat. I will grab them and if time permitting, ask them after each presentation. I have a question for you. I'm just curious, how long does it typically take for a study like yours to then be implemented into something that can then affect the public and actually be used?
Fantastic question. So that is where the drug repurposing comes in, because many of the initial IND safety, many of the things that the FDA would be looking for, we might already have answers for those. That a typical, start to finish pipeline can be anywhere from 5 years to about 7 years. To bring a vaccine to market, typically in a non-COVID situation, if all other things were equal, it might take anywhere from 7 to 10 years easy.
Thank you.
Thank you, everybody.
Well, thank you very much to Dr. Narayanan. We are now going to move on to our next speaker.